Friends, how are you guys doing? Thank you very much for joining in for one more stream. Today, once again, me Sourajit back here with another interesting topic. In the previous video of yesterday, we talked about monohybrid cross. So I thought, why not also do a video on dihybrid cross? Monohybrid cross is comparatively easier. बहुत अच्छे से कर सकते हो आप. एक बार आपने देख लिया वीडियो इंस्ट्रक्शन समझ लिया तो it's very easy. You can do monohybrid cross much easier and with different varieties of alleles as well. Dihybrid cross is a little difficult. Dihybrid cross में थोड़ा difficulty होता है because there are multiple set of alleles here or genes you can, because of which the entire calculation also becomes much larger. So for that reason only, I thought I'll try to simplify it for you today. Thank you very much for joining in with us. If you don't know what we do here at ODA Class, make sure you go and check out our app. We teach the most innovative way possible in India right now. We take care of millions of students every year, and you can be part of the team as well. I would love to see you joining for our 1 week course, which is the second best course that we have. This course is for 1 week, and it will make sure that not only you get all the help you need, but you get all the material that you need as well. This will be for 6 days, where 6 different topics and subjects, not six different subjects, but different topics from PCMB will be taught to you by different teachers as well. These teachers come from NIT IITs with more than 5-6 years of teaching experience. ये teachers आपको घर के बगल में नहीं मिलने वाले हैं. Accessibility is a big problem in India for very good teachers. And because our classes are live online classes, you can access them from anywhere. Just take out your phone and enjoy the class. Not only that, you will also be given a personal teacher. Personal teachers are the best features, including our NIT and IITN teachers. Personal teacher can actually help you with your assignment, clear out your doubt, which is a big problem. Have you ever felt after school class or something else, you have a lot of doubt in your mind and you don't know how to take care of them? The personal teacher can take care of it. Not only that, in the application, you get everything starting from your homework, test, as well as books in PDF format. Everything in one solution app itself, which is ODA app. So go check it out. If you want to enroll for this course, it's a very easy process. All you have to do is go to the link in the description of this video and enroll yourself for such a steep, low price for an entire week. You are going to pay 29 rupees, but you can gain more than 20 rupees out of this. How to know this? Make sure you wait till the end of this stream. Now, today I will be your teacher for this class. My name is Sourajit Das. You can call me Jeet sir if you like. That's how most of my students call me here. I've been teaching for more than 6-7 years now, and I teach thousands of students in our classrooms. I love teaching biology because I'm a microbiologist as well as a molecular biologist. So today I bring to you one of the most difficult topics that comes for 10th standard biology, and that is dihybrid cross. Now, dihybrid cross is one of the most sought out question in your 10th standard as well, remember. Okay, this question, every board exam I've seen, like 90% of it, this question will come. Usually they will ask you, perform a dihybrid cross with selected set of genes, or they will give you a set of genes. You have to figure out the phenotype and the genotype. Now, doing the cross is very easy, but finding out the ratio of phenotype and genotype, that's where you will do a lot of mistakes. So that's what I want to rectify today. So, shuru karte hain. First of all, let's understand what is dihybrid cross. Dihybrid cross has the same definition as monohybrid cross, just one thing is different here. Monohybrid cross deals with a cross between hybrid parents who contain two different versions of the single gene, or you can say two different alleles of a same gene. So you're working with one pair of different allele. Here, in case of dihybrid cross, you're going to work with two different sets of allele from two different genes. For example, in case of monohybrid cross, you might work with seed color, let's just say. Mendel worked on pea plant seed color and green color gene and yellow color gene. You do a cross between this and find out what will be the ratio of yellow color seeds and green color seed production. In case of dihybrid cross, you have to take one extra pair of gene. For example, let's not only take the color of the seed, let's also take the shape of the seed, wrinkle seeds and round seeds. That's what we're going to do today. So dihybrid cross is basically the cross between two organisms with different alleles of two different genes, not one gene itself. Okay, so we are working with 4 genes, but different versions of them. Now, before you do any kind of cross, these are common indicators that you have to remember. For example, whenever we write P, it means parent. Whenever we write G, it means gametes. Whenever we are writing F1, it is first generation, and F2 means second generation. You also should be familiar with these two terms. Homozygous basically means pure breed individual, means they contain the identical allele for that gene. Okay, they contain the same version of that same gene in each of the parent. For example, a parent has green color gene, both the genes are green color. Okay, no green and yellow mixed up. On the other hand, heterozygous basically means hybrid. Hybrid individuals will have two different versions of the same gene, not the same version of the same gene. Okay, two different versions of the same gene. To understand this better, let me show you some of this. You will have a better idea. For example, first step in doing a dihybrid cross, you have to choose homozygous parents. That's going to be the parent generation. For example, I have chosen here this homozygous parent. Can you see? There are two different characteristics here. One is the color, which is green, and one is the shape, which is wrinkled, isn't it? So I have chosen a homozygous parent who has wrinkled green seeded nature. On the other hand, I have chosen another parent who has the different versions of both these genes, but pure version again. No mix up. As you can see, both of them are round. Both the genes contain round gene. Both the genes also contain yellow color seed. Okay, so they are again homozygous, round, yellow seeded parent. Now what we are going to do next is the cross. Now, before you do the cross, try to assign an alphabetical letter to these characteristics, because remember, with our basic idea of Mendelian genetics, after the 2 laws of Mendelian genetics, we also derived a third law, which is the law of dominance, that whenever multiple set of alleles of a single gene is together in a parent or any other organism, one of them will strongly express itself compared to everybody else. That is the dominant allele of that same gene. Whereas some of them, which could not exceed the expression of the dominant one, they will all be called as the recessive allele of the same gene. For example, here we know that this is homozygous in both the cases. So what are we doing? We have given the recessive term for them. Small y means green color. Small r means wrinkled seed. Whereas capital Y stands for the dominant yellow color, which is the dominant version of this gene. And capital R means round, which is the dominant version. Now, just to get you a little bit clearer on this, focus here. When you see, you can understand that capital Y gives you yellow seed all the time, whereas capital R will give you round seed. Okay. Small y stands for green seed, because instead of small y, if you write capital R or capital G, it gets really confused. So first you name the dominant gene as any of the letter that you like, A, B, C, D, whatever you like. And then the small letter or lowercase of that same capital letter can be used as the recessive gene, because here we are working with pairs of gene, isn't it? So it's easier to understand. And small r here becomes the wrinkle seed. So we have both the dominant version right here and both recessive versions right here. Now, when we are taking the recessive here and dominant, both the genes here, and we want to see what gametes will be produced, based on our understanding of the law of segregation and independent assortment, we know that only one type of gamete can come from these homozygous parents, because they don't have a combination of genes. It's the identical version. So they can only produce one single combination only. Okay. Now, when these two people are going to combine and give birth to the hybrid for the next generation, which is your F1 generation, as you can see, this is a hybrid. Not only it contains both the recessive and dominant allele for that yellow color seed, it also contains both dominant and recessive combination of the round seed as well. Now remember this here, because both yellow and round seed is the dominant version, the offspring will actually show yellow color. Even though their genotype is different, the seed's phenotype will be yellow, because yellow is the dominant one. It will express very strongly, and R is the dominant one, so the seed will be round, not wrinkled. Now what we're going to do in the next step, we're going to take two of the individuals from this hybrid generation, and then we do the dihybrid cross. One hybrid, two hybrid, dihybrid. Also, one pair of gene, second pair of gene, dihybrid. Okay, so we did a cross on them. Now, in this case, a lot of gametes will produce. Now, I cannot just write them on the board, so I have a Punnett square right here for you. So if you take these, first combination can be that both the dominant alleles are in the same gamete, one dominant, one recessive, one dominant, and both recessive. Same thing will follow the same fashion in terms of the other parent as well. Now is the fun part. All you have to do is get the combination in the table. That's it. Just do the combination. This is the easiest part that you have to do. Just be very wary and alert about what combination you're writing down. All you have to do is, for example, capital R, capital Y, capital R, capital Y. I combine both of them, it becomes double capital R, double capital Y. Very easy to do. Next step is where you have to count them. Now, next step is where you usually do very, very wrong, specifically because the counting part is where you have to be very, very careful not to count them wrong. First of all, let's do the phenotype part. Phenotype part is the easiest because phenotype means the expression part. Okay, seed might have different type of genetic variation, but the expression that comes out is based on what is dominant and what is recessive. That's the reason why you will get an easier time calculating the phenotype. For example, here only phenotype that you can see is yellow color seed, green color round, yellow color round, green color wrinkled, and yellow color wrinkled. 4 different types of seeds, isn't it? So I have named them right here as well. All you have to do now is count them. Let's count them. If you want, you can pause the video and you can count it on your own as well if you want. Now, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So, how many yellow round seeds we have? 9. That's 1, 2, 3. How many green round seeds we have? 3. 1, 2, 3. How many yellow wrinkled seeds do we have? Again, 3. And the last one that is remaining is the wrinkled green one, which is 1. So what will be the phenotype in this case? The phenotype will turn out to be 9:3:3:1, which is right here. This is what you have to establish in exam, by the way. तभी marks मिलेंगे, नहीं तो नहीं मिलेंगे. Now, the genotype part is even more difficult. Why? Because इसमें देखो कितने सारे variation है. Phenotype तो जो बाहर दिख रहा है वो calculate कर रहे हो आप. But when it comes to genotype, you have to calculate every single combination and how many of these combinations are there in the table. So be very careful because this is where you will do the most amount of mistake in calculation. Now, before you go ahead, try to go through the table and list down all the combinations that are possible. Look here, I have already written down all the combinations that are possible through these gametes. All I have to do now is count. Okay, you have to count. For example, capital R, RR, capital YY. Now you have to find out, is there any other capital RR or YY? Very difficult to find one more because only 2 of these gametes can combine here. So that means this is going to be 1. Same, small r, YY. This is also going to be only one combination. There is no other combination possible. So small r, YY, this is going to be 1 as well. So take your time and try to calculate this. Just for the shortage of time, I'm showing you what I have calculated already. So this guy is 1, then 2, 2, 4, 1, 2, 1, 2, 1. So what will be your genotype? Your genotype will be 1:2:2:4:1:2:2:1:2:1. I know it sounds like a rhythmic movement, but this is what you have to remember. अगर आपको लगता है कि आप calculation में गलती करोगे, इसको रट्टा मार लो आप. Remember the ratio. So even if you do calculation mistake, at least you can verify it with your ratio when you're writing it down. Okay. If it doesn't match the ratio, that means कुछ तो आपने गड़बड़ किया है. Calculate it all over again. That's how you can do a dihybrid cross in a very easy manner. So the conclusion that we can give is that a dihybrid cross produces the offspring with phenotype ratio of 9:3:3:1 and genotype ratio of 1:2:2:4:1:2:1:2:1. That's how you have to write it in your exam. Once you finish this part, you're definitely going to get the full marks. There are other ways of getting full marks as well. Of course, I've just taught you one small topic. You can learn more of these. Our teachers are really experienced in terms of teaching all this important information. And our app is even better. So go check it out. You can also join for our 1 week course, which is going to be a life-changing experience for you. Not only because you get to be taught by IIT and NITN teachers, which you're not going to find around your house, you're also going to get a personal teacher who will take care of your doubts and help you with exhausting amount of work. Trust me, having a guiding light while you're going through the education life all by yourself is a huge help. Of course, you're going to get the material and all the things that you can possibly imagine and need as a student. Now, remember I told you that the course fee that you're paying 29 rupees here can be actually earned back, and that is through what? That is through our earning program. You can be an ambassador by studying with us and earn through that as well. We give you these coins called ODA coins, which can be earned through attending classes. You attend classes, answer questions, do your assignments. You also submit your normal tests that we usually take. All of these tasks, if you finish them, every task gives you set of coins. You can collect these coins and you can actually buy interesting gadgets for free. You don't have to spend any money. We even deliver it to you for free. Don't believe it? I gave you the information already. All you have to do is go and enroll yourself for our bridge course. The link is in the description. If you want to learn like this even more, subscribe to our channel, give us a like, and also make sure you join for our bridge course so that you can actually experience how we teach and what we have for you in the store. That's all for today. Thank you very much for joining in. I wish you have a wonderful Sunday. Take rest and start again brand new on Monday.